We're going to get started with the bell work. Number 1, we're going to do in the chat, and number 2, I have a poll for to see if you guys remember. So for number 1, we got 2 processes that we've talked about. We talked about replication and we talked about transcription. And each one, the base pairing rules are a little bit different. So who remembers the base pairing rules for replication? What goes with what? Yep, Desiree got it. She's first. A goes with T and C goes with G. So if a question asks for replication or asks for a complementary DNA molecule, you're using the basic Chargaff's rules, A and T and C and G. And what about transcription? What's different? Good, Angelina. A goes with U and G goes with C. What about T? If you see a T, what does that still go with though? An A. Yep. So T with A with U, and then G's and C's stick together. Good. All right. So we are going to do a poll. So for 1A, if I ask you, what is the matching or complementary DNA strand for CAA, GTA, and GGC, what are you going to pick? And there's a reason we group them by 3s, which we'll find out today. So for part A, I'm asking you for the complementary DNA strand. So this would be replication rules. Give you guys a couple minutes to answer these questions. What pairs with CAA, GTA, and GGC? Oh yeah, you're right, Angelina, or Desiree. So number 3, the answer is actually not technically up there, but most of you are picking the right one. I gotta fix that. I'll give you guys 30 more seconds. To answer. Hi, Jamie. Hi, Anthony. Hi, Christopher. Hi, Alex. All right. So I'm asking you for the complementary DNA strand for number 2A. So CAA pairs with GTT. The majority of you got that right, but this is complementary DNA, so we're still using T's. Hi, Demetrius. GTA pairs with CAT, still using T's. And then GGC should pair with CCG for number 3. But most of you answered that one correctly. That was the closest one. All right. Now I'm going to ask you for the complementary RNA strand. So same thing. CAA, GTA, GCC. But now what is the RNA that goes with that? I told you guys last week, you have to pay attention to what the question is asking for. For 2A, I was asking for the complementary DNA. For 2B, now I'm asking for the complementary RNA. So what letter should I see in the RNA that I didn't see in the DNA? Most of you are getting the hang of it. Give you guys another 30 seconds or so. All right, so we learned from our mistakes last time. CAA now pairs with GUU. Remember, if I'm asking for RNA, I should see U's instead of T's. GTA pairs with CAU. And then GGC pairs again with CCG. That's not there, but you guys chose the closest one, which is fine. All right. So normally we break 8.5 up into 2 parts, but we're doing them all today. So for this first part, we're going to talk about translation, and then we'll switch over and talk about the genetic code. So essential question, what are the steps in the process of translation? We've broken it down into a 3-step process, just like we did with replication and transcription. So translation converts the message encoded in mRNA into a protein. So last week we talked about transcription and how the information in DNA got transcribed into an RNA molecule. That RNA molecule is mRNA, messenger RNA, and there's different types of RNA that you guys should have learned in your assignment last week when you were reading through the article and answering the questions. And the first thing we're going to do is we're going to go over some of the pieces that you see. So you got to know the pieces, what they are, before we know how to put them together. So one piece is a codon. A codon are nitrogen bases grouped into 3s on the mRNA strand. So last week, transcription made an mRNA strand. And every time we talk about transcribing the RNA, or even when we do a complementary DNA molecule, we always separate them into groups of 3. And this is why. It's the groups of 3 are very important. And then each codon gets translated into a different amino acid. Remember, the amino acid is the monomer, or the single subunit of a protein. Put a bunch of amino acids together in a big long chain, you get a protein. Another piece you have to know to understand translation is an anticodon. So the codon on the mRNA strand attracts its complementary bases based on our rules. Remember, we're still talking about RNA, so A and U are going to pair together. And then G's and C's are going to stick together. And the complementary bases are called anticodons. And they can be found on a tRNA molecule, which is the next piece we'll talk about. Make sure you draw this picture. On the bottom, you have codons from an mRNA molecule. On the top, the nucleotides that pair with it are called anticodons, and they're found on a tRNA molecule. And the anticodon is not the only thing found on a tRNA molecule. So this is what a full tRNA molecule looks like, and the T stands for transfer RNA. I think it looks like a little hunched over old man. And at the top, his little head is an amino acid. And at the bottom, that's where the anticodon can be found. And that is what will seek out a codon on the mRNA molecule. You'll have questions this week on your attendance quiz and on your Check for Understanding quiz. Where can you find an anticodon and where can you find an amino acid during translation? And they're found in the same place on the tRNA molecule. All right, so now that we know the different pieces, we're going to go back and just review the last step of transcription really quickly. The mRNA strand, which is the red one. In the end, it leaves the cytoplasm to go find a ribosome. So that's the end of transcription. The mRNA strand just goes away. And its ultimate destination is outside into the cytoplasm to find a ribosome to complete this three-step process. So translation, we have 3 steps. I put all 3 steps on the same slide. Step 1, the tRNA and the mRNA come together in the ribosome according to their base pairs. So if you look at the GIF, the big pink blob in the background, that's the ribosome. And the pink, bright pink strip at the bottom, that's the mRNA, and then that tRNA is the molecule in the middle. Looks a little different than our little hunched over old man, but it's the same. It has the anticodon on the bottom, and then amino acids on the top. Step 2, the amino acids bond to each other on the top. And then step 3, the result is a long chain of amino acids, which make up a protein. So this GIF is kind of the overall process, all 3 steps of what's happening during translation. Finds a ribosome, the mRNA and the tRNA come together according to their base pairs, the codon and the anticodon. The amino acids bond to each other on the top. And in the end, you have a long chain of amino acids, which is what a protein is. We'll watch it one more time while people are finishing writing. All right. So now we're moving on. Part 2 is about the genetic code. So the question is, how do you interpret amino acids from RNA molecules? How do you know which amino acid goes with what? And the way proteins differ from each other is dependent on the number and sequence of amino acids that are translated from our DNA. 2 weeks ago, we started in the DNA. Last week, we learned how to transcribe it into RNA. This week, how do we translate it from RNA into an actual protein? All right, so you have to read the code. So how do we know which codons match which amino acids? The code is a chart of all 20 amino acids found in living things. And that chart looks exactly like that. That's not something you're going to have to memorize, but you do have to learn to interpret it. And it's actually very simple. You can see in the middle are abbreviations of a bunch of different amino acids. PHE at the top left stands for phenylalanine. Below that, LEU stands for leucine. So each one of these is an abbreviation for an amino acid. And there are only 20 amino acids found in all living things. And you think that's only a little bit of information, 20 amino acids to code for all of our proteins. But that's what happens. So it is the order of these amino acids that determine which proteins are expressed. So if you think of the English alphabet, we only have 26 letters in our alphabet, yet we have millions and millions of words. How? It's because we rearrange those letters in different orders and different lengths. Some words are only 1 letter long, 2 letters long, but some words are 15 or 20 letters long. It's all about the arrangement of those 26 letters, just like it's all about the arrangement of these 20 amino acids. So for example, we're going to use the codon AUC and translate it into an amino acid. So step 1, you take the first letter, A, and you find it on the left side of the table. So you can see in the picture, first step is pointing to the A. Now you have a row to look in. So your amino acid for AUC is going to start somewhere in that row. Step 2, you take the second letter. Our second letter is U. The second letter is found on the top of the table. So now you have a row and a column where the A and the U intersect. Where they intersect, there's a box. And then the third, you only got one more side, you find on the right side. So the third letter in our example is C. So you go to the right side, you go down to your row that you already established, and you point to the C. And that C will indicate where in your little box where the A and the U intersected, where you're supposed to be looking. And you can see the AUC there. You won't always be able to find AUC in those little boxes. Sometimes it's just the name of the amino acid. But for now, while you're learning it, you can see it. So you just have to figure out where all 3 of those letters intersect. So AUC codes for ILE. That's the abbreviation for the amino acid isoleucine. And like I said, this is not something you're going to have to memorize. You don't have to memorize this chart, but you do have to know how to read it. And if I give you 3 letters, how do you read the chart to figure out which amino acid comes next? So we are going to practice because you do have questions on it this week. And here's the summary straight out of your book. This is on page 224 in your big green biology book if you have one. So the genetic code matches each mRNA codon with its amino acid or function. And the example they have here, suppose you want to determine which amino acid is encoded by the CAU codon. So our first base is C, you can find that on the left. Second base is A, you can find that on the top. You figure out where C and the A intersect. And then the third base is U. CAU points to the top amino acid in that box called histidine. So try it. I want you to interpret GGG, CCC, and UUU. Let's see how you do. You got your first base on the left, second base on the top, third base on the right. And these are the kind of questions you're going to see in your attendance quizzes this week, as well as on your test or on your quiz, and then on your big test in a couple weeks. So you take them 3 at a time, which is why we always separate them by 3s. G, G, and G. C, C, and C. And U, U, and U. I'll give you another 30 seconds or so. Most of you are getting it. There's a couple people who are a little confused. That's okay. All right, so most of you got the first one, GGG. So we have G on the right, G for the second one. Where do those meet? They meet up in this box. And then the third one, you have G on the bottom, which is glycine, GLY. Number 2, you have CCC. C and C. They meet up in this box. There's actually only one choice, but C, C, C. You got the C. Points to proline, PRO. And then U, U, U. There's U and U. You got your first box where they meet up right here. You got 2 choices in here, so you got to look at your third base, which is, there's the U. Points to the top one in your box, which is PHE. Stands for phenylalanine. All right, like I said, you're going to see a lot of those questions in the next couple weeks, this week, next week, the week after. So hopefully you know how to do that. If you don't, feel free to ask questions. I will help you if you didn't quite get it. But in the end, you end up with a protein. You're going to end up with a chain of amino acids. Amino acids are the monomers. You put enough monomers together, you get a polymer. And a protein is that polymer. And then there's a repeat. There are only 20 amino acids that determine all of our genes. Like I said, think of the alphabet. There's only 26 letters, but it can make millions of words based on how many letters are in that word and the order of the letters. So our genes are determined by the number and sequence of amino acids that are translated by the three processes: replication, transcription, translation.